From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 43, recorded on May 22nd, 2019. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels Elde.
Greetings, Vincent. Good to be back. I was just back on the East Coast in your neighborhood, and flew back last night. I was in Philly, kicking myself. I should have just tried to extend my, flight for another day. I could have come and joined you in vivo with our in vivo guest today.
Well, it's okay. I understand. You could have taken the train up and back, fly out of Philly and so forth. Mm-hmm. No worries. So we do have a guest for you today, right here in the TWiEVO Studio East. Is what it is right now.
That's right. Elde Lab Studios is TWiEVO West.
TWiEVO West. He's right here at Columbia University in the Department of Ecology, Evolution, and Environmental Biology. Dustin Rubinstein. Welcome to TWiEVO.
Thank you. It's great to be here. Easy commute.
Very easy, yes. Although, I don't know how you got here, but the one doesn't stop at...
I know. I took the campus shuttle, but it's graduation today, so that was a little bit delayed
crowded,
too.
Yeah.
So.
A lot of people walking around.
Yep.
Yeah, today's Wednesday. It's graduation day. And we've had...
This is for all of Columbia?
Well, Morningside, right?
Everything was today. It was the big ceremony with the medical school and everything.
Medical school, too.
Yep.
I saw somewhere that they had some ceremony at Radio City this week for one of the schools.
I think Barnard College does their graduation down there, and then each of the schools does their specific graduation at some point. But today was the big one with everybody, and the president speaks.
So, do the medical students also go to Morningside for that? Yeah,
they do. Yeah,
because I know they have their own ceremony up here, and they have.
Yeah, this is the big one where they take the whole center of campus and they decorate it, and security locks it down.
I did that once. I was what do you call it, an usher? Whatever it is that the lowest thing a faculty can do.
But you were wearing your regalia?
I had to wear my gown with a what do you call it, a hood, which is from the place where you got your PhD. Mm-hmm. So I had to rent one because I don't have one. And I basically told people where to sit. And people said, is that why you got your PhD, so you could be an usher?
But then you get to sit on the stage afterwards.
Yeah,
right?
you get to sit. It was fine. I didn't mind. It was cool. And if they have a good speaker, who do they have this year, Jim?
I don't know. I didn't go today. I don't know who it was, but it was a nice day for it.
Yeah, it is beautiful out. Did you have graduation there, Nels?
Yeah, it's been happening here. They kind of split it up into separate days. I was just thinking, I missed my PhD graduation. This was at Chicago. And, there was an unknown, at the time, speaker who was there for the commencement, and it turned out it was a state senator by the name of Barack Obama.
Oh my gosh.
Oops, probably should have showed up for that one.
You never know, right?
You never know.
Oh, that's awesome. I saw somewhere they have Bill Nye as a graduation speaker. Someday, Nels, you'll be a graduation speaker.
Well, let's not hold our breath. I don't have a robe. I need to invest or rent, I guess. Rent to own? I don't know. Yeah.
Anyway, we've never met, Dustin and I, but you guys know each other, right? You've met before?
We do.
Yeah. Dustin generously invited me out to a really fun symposium. This was a couple years ago now, isn't it, Dustin?
I think it was about 2.5 years ago, yeah.
Yeah. So the topic of the symposium was social evolution and genome complexity. And, honestly, a little outside my wheelhouse, I would say. But I was thrilled to join the crew, and we'll be talking about this in some detail. There's a new review paper that's about to land in Trends in Ecology and Evolution, which is a product of the symposium. And so, whereas my lab doesn't work directly in the evolution of social behavior, I was at least able to kind of maybe just hitchhike a little bit that day. I spoke about viruses and how some of them, when they infect hosts, can change the behavior of the hosts, in some cases to favor virus replication. Some really cool stories about zombified hosts because of virus infection. This is some of these stories are echoing throughout different systems and insects and other situations. And so, but I was thrilled to be part of this, and in particular to have sort of a front row seat to listen to folks who do this for a living, who really work on social evolution, the evolution of social behavior, and are beginning to connect this to genetic mechanisms. And as we'll talk about a bit today, maybe even the reverse. How does social behavior even influence the organization of genomes themselves?
I was just I did a TWiEVO in Rotterdam, and one of my guests was Vera Ross, who works on how insect viruses change caterpillar behavior. Mm-hmm. And they make, so normally caterpillars go up and down the plants, and this virus, when it infects them, makes them go all the way to the top and stay there. Then they rupture as the virus kills them, and the virus spreads all over the plant. More efficient way of spreading the infection by making the caterpillars go up.
Yeah, I've always looked for those in the rainforest to see ants or caterpillars climbing a tree with the fungus growing out, but I've never seen them in the wild.
So let's start by getting a little of your history, Dustin. Where are you from originally?
I'm from New Jersey originally.
Really? What town?
Outside of Princeton, a small town called Pennington.
Pennington. Yeah, I was born in New Jersey and raised there as well.
Okay.
But I was born up here, and I was born in Patterson.
Okay.
Distinctly
not too far
different neighborhood than Princeton.
Yes.
But I do go to Princeton a lot to work on our textbook. So you went to high school there in New Jersey?
I went to high school in New Jersey, and then I went to college in New Hampshire.
Which one?
Dartmouth College.
Dartmouth. Oh, I just had a student yesterday who, let's see, what did she? Oh, she got into Dartmouth Medical School, which is called Geisel.
Yep.
Because that's Dr. Seuss, right?
Yep. He was an alum, an undergrad alum of the college.
Cool. Were you a science major?
I was a biology major, and I also majored in earth sciences and environmental studies.
Where did you get your interest in science from? Do you know?
My father's a professor in biology, so when I was young, I got to travel the world with him, spend summers studying animals in Africa and Middle East, and it was fun. So I got exposed to that. I wasn't clear that's what I was going to do. I was interested in earth sciences a lot in college, and I got very interested in using stable isotopes to study movements of animals. So there was still a biological aspect, but then I got away from that a little bit in graduate school and got more interested in social behavior and social evolution.
I was just up in Albany, and there's a lady there who studies animal behavior in Africa, and she just takes videos of them. And I went into her lab, and her lab is just computers, and the students are just looking at the animals moving around on the video.
Yeah, we have some of that. We're trying to go to more automated methods these days. The tools are out there where you don't need an army of undergrads to do this. It depends on the level of the complexity of the behavior that you want to score. But the hope is that the technology will catch up, and we can automate most of this stuff now.
Yeah, I'm sure it will. It's like everything else, right?
Definitely.
Yeah.
So after New Hampshire, after Dartmouth, where did you go next?
I went to do my PhD at Cornell.
Ithaca.
So
I was an undergrad at Ithaca.
Okay.
But I graduated in 1974.
Okay. I graduated in 2006.
Oh my gosh. Yeah, I'm an old guy. But Ithaca is very different. I haven't been for a long time. And
it's gorgeous.
It's very nice, yeah.
Literally.
And who did you work with there?
I work with Steve Emlen and Paul Sherman. So Steve Emlen studies social evolution in birds, and Paul Sherman in mammals. And so I worked with both of them. They're both retired now. It's
a great place to do ecology and evolution, right?
It is. It was one of the first departments that sort of brought the mechanistic side to behavior. So it brought the behavioral ecologists together with the behavioral neuroscientists in the 1960s. Thomas Eisner, the chemical ecologist who studies insects, that was sort of his vision, and he created the department. And I think it's produced a lot of students that really think about that integrative perspective and how you bring mechanism into studies of function.
I had a roommate who took a course with him. Just adored him. Right.
Yeah, he was a great guy.
Did you know him at all? Or?
I did a little bit. He was still there when I was starting. He had Parkinson's, and he was getting sick towards the end, but he was still very active right up until the end.
And then what was your next stop after Cornell?
I did a postdoc at Berkeley for 3 years before moving here in 2009.
And who was that with at Berkeley?
It was somewhat independent. I was a Miller Fellow, so I was sort of doing my own thing. That's when I started. I worked on birds for my PhD. I still work on birds, but I was interested in doing doing some work on invertebrates to do some comparative work. So I started working with snapping shrimp, and I was sort of based in the lab of Roy Caldwell, who studies mantis shrimp. They're very different from the shrimp that we study, but there's not a lot of people out there that study social behavior in crustaceans. And so.
That's right. And we'll talk a bunch more about that, but maybe can I step back for a minute, Dustin, and talk a little bit about this, being a Miller Fellow. So just for our listeners a little bit, I think we're all pretty familiar with the kind of academic path from grad school, postdoc, starting a lab. But what is this? This is sort of a hybrid postdoc, almost like you have a little more independence, a super postdoc. How would you describe that experience?
It's a pretty independent postdoc. You sort of apply with a project in mind, and preferably sort of a mentor in mind. But it's across the physical science, the natural sciences, and the physical sciences to some degree. So physics, astronomy, biology, chemistry. And they take about 10 people a year into the program, and you can kind of do whatever you want. You're given the freedom, you're given, a salary and some resources. And it lets you sort of develop your own, program as a postdoc that you can then parlay into a job. This country, as you guys know, doesn't have a lot of those independent fellowships in the way that some of the European countries do. And so it's one of the few nice ones that really lets you become, an independent PI at an early stage and get your program up and going.
And you'd do it again if you had the chance? You're happy with how that turned out?
Definitely. I mean, I look back on the postdoc years fondly when my calendar is empty. I don't have any meetings. I don't have any classes to teach. I can read. I can write. I can think. And those things are a little bit hard to come by these days. But you're with a group of, there's about 30 of them over the 3-year fellowship. And so you're with a group of really diverse scientists, and you talk and you learn about, all sorts of things. That's where I first met Corey Moreau. We overlapped for a year or two, and a bunch of people in various disciplines that I still stay in touch with. And it really challenges you to think differently about the research that you're doing by interacting with these people regularly.
So
that's great.
Did you have a project in mind already when you applied for it?
I did. I was interested in switching over to these shrimp and doing some molecular work to understand the genetic structure of their colonies. It proved challenging, but we started that, and I still work on that now. And now I have a bunch of systems, but that's one of my primary systems that I was able to start then.
All right. And so it sounds like you do field work, right?
I do less and less these days, but we do sort of two components of fieldwork. We sort of have long-term studies of marked populations of birds, primarily in Africa, and we return to the same sites and monitor the same birds, for 12 months out of the year, basically, and they live for a long time. And then we also do repeat sampling of other populations in places to do sort of longitudinal studies or to look at spatial differences in population.
In birds as well? In
birds, but we also study beetles, the shrimp. We've just put in some grants to work on some rodents in Australia and some lizards in Australia. And so we work on a variety of things that all sort of center around the questions that the lab focuses on.
And when you do the shrimp field work, where does that get done?
So it's a group of about 45 species of shrimp that live primarily in the Caribbean.
Tough one.
It's tough. So we work in Florida. We work a little bit in Panama, some of the other Caribbean islands. It sounds glamorous, but doing a lot of marine field work is scuba diving or snorkeling for an hour or two. We collect a lot of dead coral rubble and then spend 8 hours bent over a dock, picking through the rubble, looking for these shrimp, and then late into the night, sort of identifying them. So you're outside for an hour or so a day.
Yeah, no, I think a lot of people think it's a glamorous thing, but it's not. And in our field, we don't do that very much. Although there are some people who do. I've never done it. And I kind of look with awe at people in other fields who do field work, and I can't imagine coordinating it. We've talked to a few people, Nels, right, who do this, and
the logistics that go into this, and all of the extra variables, not just the scientific variables that we all train in the laboratory to think about, but then all of the things out of your control for actually navigating countries, permits, weather. It's really an incredible amount of both skill and luck, it seems like, that goes into a lot of these approaches.
It is. And one of our primary questions is looking how environmental variation shapes social living. And so we're interested in how year-to-year climate variation changes things. But that means that some years you go down prepared to do experiments and it doesn't rain and the birds don't breed. And we're in the midst of that year right now, and I have a frustrated postdoc who's not getting as much data as they would like because it hasn't rained much in the last 3 or 4 months.
Have you read The Beak of the Finch?
I have.
So it's our colleague at Columbia.
Yes.
I'm forgetting his name.
Jonathan Weiner.
Jonathan Weiner,
Should have him on some.
He was. I think he was on TWiEVO, right, Nels?
Yeah, he was. I'll try to pull up that episode.
He talked about, he's been twice. He was once on another podcast of mine. I don't remember which one, but also once with Nels on TWiEVO. We talked about
time, love, memory.
Time, love, memory. Yeah.
Yep.
The clock stuff, which got the Nobel Prize like a couple of years ago. He was very excited about that.
He was writing a book on that, I think, or he has been for a while.
He wrote a book, and then the Nobel came out a few years after the book came out. So it was kind of, and the people in the book all got Nobels except for except for Sidney. No, what's the guy's name who started this whole field, Nels?
Sidney Brenner?
Not Sidney. No, he was
Seymour Benzer.
Seymour Benzer.
That's right.
Get the SBs. Yeah, that's right.
But he started it.
Yep.
It's just a great book. Yeah, he's great guy. I teach a lecture in his course. He has a science writing for journalists. Would you teach that?
I've done it once or twice.
It's a lot of fun.
Some good students.
So Jonathan was on TWiEVO episode 19.
Okay.
And for folks.
Yeah, and I had him on another one for Beak of the Finch, but I don't remember what one it was. But he's been on twice. It's a lot of fun.
Yeah, I think that was before. Yeah, maybe it must have been on TWiV or...
I know. I had him here with Dixon. I think it was TWiV. Yeah. I also had Stuart Firestein on because he's written 2 interesting books, right? On failure and ignorance. Why can't I remember that?
Yeah, we still give Stuart's book, Failure, we still have that available to all our first-year grad students.
That's good.
And offer it to them over sort of holiday break, as it's not required, but a lot of them are taking us up on this. We talk about it in some of the classes here about how to think about science through the lens of failure. It's really useful, I think.
Yeah, it's a good book. So, Nels, you want to chat a little bit about the symposium?
Yeah, absolutely. Although I think let's finish off the history. So we were finishing up, or maybe I missed it, but we were finishing up the Miller Fellowship. And then how about the transition from there to your own lab? How did that happen?
It happened quickly with a child born in the midst of all that and moving cross country. So it was a bit chaotic. I got hired right before the economic downturn. So I arrived at Columbia in the midst of that. No lab renovations and everything took a while. So it was a prolonged transition, I guess you could say. But we've been here now 10 years.
Yeah.
So still going strong.
And just to illustrate the complexity of this place, I didn't even know you were here, right? Because we're up at the medical school. There's barely any communication between the 2 campuses, right?
Yeah. I mean, it's 50 blocks, 40 blocks different, but it could be night and day. It's different worlds, unfortunately.
It's too bad.
Mm-hmm.
I mean, they've done a good job with the neuroscientists by trying to move them down to this new campus. Yeah. And so there's more interaction now around neurosciences, but not in your field yet.
No.
Well, and I would say this is one of the reasons we do TWiEVO is to start to try to attack these barriers that are both geographical and sometimes almost science cultural. And I'm pretty happy to see on my Skype screen the 2 of you hanging out together in the same office. I think it's working for us to talk between disciplines here. I'm really thrilled.
It certainly isn't the usual way to get. I would never talk to Dustin, right, except for TWiEVO, because our fields don't overlap at all. And there's not much virology at all. In fact, nothing downtown.
True.
True. Morningside.
There's not a lot of biology compared to a lot of places, but there's a heavy density of it up here.
And that's why I started teaching a virology course 10 years ago now at Morningside. I said, this is crazy. These students don't have a virology course, and you need it, right? Half of the class is going to be a doctor of some kind, right? They need to know virology.
Yep. Yeah, the students are the bridge. I mean, I'm sure you have lots of undergrads that are coming up this way and working in labs. They're good about, finding ways to get up here, but the faculty, are relatively siloed among the schools, unfortunately.
Having a course really helps because then they know, because other than that, it's hard for them to figure out. And they're only there are only certain faculty who are anointed up here to be able to take PhD students, at least from downtown. But other students don't find you unless you teach a course. So anyway, so then you're here 10 years. Whoa.
Yeah, time flies.
Time flies.
You're 35 years.
Time really flies.
It really does. Yeah. So Nels, conference, you want to talk about that now?
Yeah, so maybe remind me, Dustin, how this came together and kind of your thinking as you were organizing and running that conference. It was like a day or so together. Actually, more than just the symposium. It was also a kind of micro meeting.
Yeah, so this was the brainchild of Hans Hoffman and I, and we had been thinking about these ideas for a while, but we both study social behavior from different perspectives. Me more from the behavioral side, him more from the neurobiological side, but we're both getting more genetic. And there's a huge emphasis on sort of the genetics underlying social behavior, but it's really thinking about, the genes and the mechanisms that drive individuals to become social, and the things that cause species to adopt complex social behaviors, complex social systems. But we were kind of interested in the flip side of that, as you mentioned, thinking about what the implications of being social have on the genome itself. And so the goal was to bring a lot of people together that don't always speak and that don't always know each other, like bringing you in, a virus biologist, to think about genome evolution. And I think we succeeded in that. We really thought about new things. And so we had one day of sort of public symposium. So this was funded by the university seminars here, which was set up, I should know this, about 80 years ago. It gets actually groups of faculty and community members interested in topics, and they meet, once a month or something like that over dinner and have seminars. And then they also sometimes sponsor these symposiums. And so we have a university seminar in the integrative study of animal behavior that we created a few years ago. And as part of that, we got some funding to put this together. And then Hans was able to leverage some money from an RCN NSF grant on sort of sociogenomics. And I was able to bring in the speakers and put them up. And so we had a day of public talks where everyone could, get on the same page and hear about what we were all studying and what we were all thinking in this area. And then we kind of locked ourselves in a room for 2 days, I think, and talked and thought about this paper, and then sketched out the paper. And then it sat for about 2 years, which is probably my fault and Hans's fault. I was writing a textbook in the meantime. And when I finished that, we got back to the paper. But I think that gave us time to have perspective, and some new stuff had come out. And we were able to put this review paper together. But it was, I think, a really forward-thinking review, because there actually isn't a lot of data on the sort of the genomic consequences of being social, how living in, family groups or having complex mating strategies feeds back on the changes in the genome.
Totally agree. And so just to note that Hans Hoffman, his outfit's located in Austin, Texas. I think, do you remember, is he in the Department of Integrative Biology there?
He is.
Oh, okay, yeah, yep. And also Institute for Cellular and Molecular Biology. And so the two of you really stepped up and spearheaded this. Some of the other folks that were involved have been have shown up on TWiEVO, actually, and I wanted to highlight that. You already mentioned, Dustin, Corey Moreau, who you collided with early, I guess, in your Berkeley days, when you're a Miller Fellow.
Yep.
So she was on TWiEVO episode 13, This Week in Ants, we called it. And if I'm remembering right, Vincent, you actually wanted to start a podcast about ants because you're so taken by the incredible biology and just all the topics that could flow from that and connect to people as a potential podcast.
Yeah, I actually wanted to be broad, right? Entomology in general. So I asked her if she wanted to do it. And after a few months, she said no. She would be too busy to do it.
I saw on Twitter today she just had a paper on viruses in ants and how they influence genome evolution. So
really?
You can have her back for number 45.
Oh, that's a good idea. Let's see.
Like
look it up.
And now she's moved. She's at Cornell of all places. So.
Really?
That's right. She was at the Field Museum and University of Chicago, and then moved to Cornell. That's right.
Let's see if I can find it quickly here.
And then another, while you're doing that, Vincent, I'll keep running here. So another one that we had was Hoekstra came by and was a great guest on TWiEVO. This is episode 15. And I think that episode is something like From Cockroaches to Grizzly Bears, as she was sketching out her kind of illustrious past and going from organism to organism. She is now at Harvard and chair of organismal and evolutionary biology. And I think runs mammals at the museum there. Is that right?
I think she's the curator of mammals at the museum.
Yeah. Mm-hmm. So also really cool, social evolution, using some of the mating strategies among mice and building some of these crazy enclosures, kind of bringing the scientists back out into the field. So we're bringing the laboratory to the field in a sense, with some really interesting work there. So it was a really great crew of folks working on diverse stuff. Who are some of the others, Dustin? Let's maybe go through the list here. So we had
some social insect people. We had Amy Toth, who studies wasps, sort of the genomics underlying social behavior in wasps.
That's right. Karen Kapheim as well.
Right, who studies bees.
Mm-hmm. She's at Utah State, sort of in my neighborhood. Logan, Utah. It's about an hour north of Salt Lake City. Lots of work on, again, of course, on insects. Not too surprising that this would be a hotbed for that. And Amy, is she at Illinois? For blanking at the moment. Iowa State University in the Midwest. Yep. And then LaRon Keller, is there another major player in the insect?
Another ant person.
Social evolution. Yep. Lucia Carbone, who's at OHSU, she studies gibbons mostly, with a focus on the transposable elements. This kind of, I would say, emerged as a real theme for this notion of how, as Dustin is saying, how social behavior might almost feed back into genome structure. What are the mechanisms underlying this? Mm-hmm. Transposable elements, as we've talked about a lot, Vincent, and the roles they play as sort of a diversifier, sort of the fuel of evolution in some cases, right?
Yeah. But that's the opposite of the way I think of it. I always think the genome changes and then that influences what organisms do.
And that definitely happens. And so,
for sure.
So we wanted to think about the flip side because it's underappreciated in many ways.
But you say there's not a lot of examples of that, right?
There are some examples. I mean, I think the perspective we took in the paper was thinking about, how the sort of the population genetic structure of a species that lives in groups and has subdivision and not all individuals get to reproduce, what the implications of that are on the genome, how that might lead to new pathways and new genes, things like that, but also constrain things in certain ways with low effective population sizes and things like that.
Mm-hmm. Could also, we talked about this a lot, was how. So as you're setting up sort of social structures, and then what does that mean? I mean, you're kind of getting at this as well, for things like gene exchange or limits of gene exchange, or selecting on certain events, genomic changes that then sort of get locked in, so that there's this idea of the supergene that's come out from a lot of the ant studies, where you have these inversions that then lock in sets of genes or very large genetic regions. And it appears that this is I'd say a hotbed of research today, is how common is that as a sort of outcome of potentiating social behavior in some cases.
Definitely. There's two good studies in ants showing that there are these supergenes that they call social chromosomes that underlie major differences in social structure. So some ant populations have colonies with a single queen, and some have colonies with multiple queens, and we see this repeatedly across a diversity of social invertebrates and vertebrates. But these two studies in ants in particular, identified this chromosomal inversion that underlies these differences. And similar things have been seen in birds. So the ruff, where there's a species that males have three alternative mating tactics, a territorial male, a sort of sneaker male, and a female mimic. And these also have been tied to two independent chromosomal inversions. And I think, one of the points of this paper is that these differences in social structure, these differences in mating strategies in many species, these inversions or some of these other mechanisms we talk about in the paper might underlie this. And now with full genome sequences at the chromosomal level and the ability to detect these inversions, we were sort of hypothesizing that these things may occur more commonly. And it'll be interesting to figure out in cases where there is a genetic mechanism, and other cases that may be sort of more environmentally driven. But the new genomic tools will allow us to sort of test this and generate a whole new set of hypotheses and studies to sort of get at that.
So the idea of the paper was to say, okay, here are all the things that can change, and so go do it.
Exactly.
You wanted to stimulate.
Yeah. So as Nels said, there's some evidence in these sort of social species where we have good data, and others not yet from the social species, but we know that these sorts of mechanisms affect, phenotypic diversity and whole sorts of traits, and behavioral variation is one of those traits. And so it was sort of almost like a roadmap and a call to arms, also saying like, these are some areas that, are going to be fruitful for future study. We have the tools. We need more theory. We need more, and to do that, we really need to get people in the room that come from different perspectives that study this in sort of a non-behavioral perspective and bring it to the social behavior, which is a relatively complex trait compared to a lot of things that people look at.
So there's a great table here where you have these genome architectures and then any known examples, right? When you got your shrimps there and salamander brain size, odorant receptors in ants. And then there's some unknowns where there's no examples of where, say, chromatin folding has an effect. But there's one that's interesting to me, organization of genes, gene order, which you have unknown. Mm-hmm. But, in viruses, if you mess with gene order, it can screw them up.
Yep.
There's some good examples of that with RNA viruses. You can shuffle the genes and they still will work, but they're very sick and they would never make it in nature. So that's interesting.
Yeah, agreed. So, and just to backtrack for 1 second. So the paper that we're talking about is, it's not in the wild yet. So it's just about to get published. It was accepted very recently in Trends in Ecology and Evolution. And the title of the review is Coevolution of Genome Architecture and Social Behavior. And as soon as it comes out, we'll update the links so that folks can get to it. It is going to be paywalled because this is a Cell Press situation, but if you can't get access to it, don't hesitate to reach out to us and we'll get a PDF copy to you.
I think they said it'll be out in August. So it'll probably go online as a up and coming paper in the next 2 to 4 weeks, I would think.
Yep. So the kind of preview version of it.
Yep.
So maybe Nels, our goal will be to get all these people on TWiEVO.
Yeah, we're off to a great start. So we've got Dustin today, we had Corey, and we've had Hopi. And actually each one of these authors could tell an amazing, cool story.
Sure.
Let's see. So we mentioned Lucia, Hopi, Karen, Laurent, Corey, Amy, also, and Hans Hoffman, who is the other kind of co-organizer of all of this. And then 2 others, Arvid Agren, who's in Sweden, or actually he might be.
He's at Harvard now, I think.
Harvard. Yeah. Yep. And he brings in some of these ideas about genetic conflict as it relates a bit potentially to social evolution. He also thinks really deeply about population structure, quantitative approaches, including with transposable elements. And so that he had a really nice contribution as well.
Yeah, he was one of our sort of theory people to bring in and sort of ground us in some of that.
I remember when you went to this, Nels.
Yeah. Did we stop by and do a TWiEVO?
I think you did. This is 2017 now, right?
Yeah, that sounds right. Yeah, exactly.
So I just tweeted Corey these endogenous elements in ants. I said, let's get you back on TWiEVO and talk about this.
Yeah, good.
So,
any response? Twitter works like
I try and keep off Twitter while I'm podcasting. Otherwise, I won't pay any attention to what we're talking about.
It's probably a good policy
because it's very distracting. Because you see all kinds of interesting stuff popping up, right?
Oh yeah,
you get distracted. Right. You can put. Last week, I unfortunately got involved with a discussion with an anti-vaxxer. That was pretty much a waste of time because
on the podcast.
Yeah, no, it was. I was waiting. My wife was in surgery, and I was waiting for her to come out. Right, so I was grading my final exams, and I had Twitter, and this thing started going, and it was just a waste of time because in the end, you can't get anywhere. And in the end, this lady said to me, "I don't believe anything you tell me."
Yeah.
Right.
So these are not conversations. These are.
Yeah. She said, "I'm not talking to you anymore." And I said, "Well, we never did have a conversation, right? Because you're just telling me you don't believe anything I say." So it's really sad because I read yesterday that this is going to become an election issue for 2020 vaccine rights. Isn't it crazy?
It's nuts.
Totally crazy. Anyway, that's why
I had a student once who, when I was a grad student, I was TAing animal behavior. And, the whole semester, she did her assignments and went through the whole thing. And then on the back of her last assignment, she wrote, I don't believe in evolution, and so I don't believe in anything that I've written the entire semester.
My gosh.
Well, and in the end, you don't have to believe. I mean, this isn't belief or this, right?
No,
I think
she
it's the
went
wrong
through the
question.
exercises. Hopefully, she learned something.
Yeah.
I don't think it changed her, but maybe it'll change her.
It's not a belief system. We had a guy on TWiEVO. We were talking about how you can date how old viruses are by looking at endogenous viral elements and genomes, right? Mm-hmm. And there's a nice paper we did where showed retroviruses were around 450 million years ago, Ordovician, right, times. And this guy wrote, and he says, you're testing my ability to believe that this is not congruent with what I think. So he said, can you help me? I said, well, look, these are the data. We are not, overinterpreting. We're not making things up. This is what the sequences tell us, that these things were around 400 million years ago. And, I can't do much more than that. I respect what you believe in, but the science is the science. It's
it's a scientific method, right? I mean, that's what we apply as scientists. And, I teach in what's called Frontiers of Science class here. So all Columbia students take a core curriculum, and about 15 years ago, this was the anniversary actually, they introduced some science into the core. And so they get, lectures on neurobiology, evolution, physics, and earth science, climate science currently. Mm-hmm. And, one of the goals is basically to have them think scientifically, right? Sure. I mean, whether you're going to go into a science career or not, you need to evaluate evidence when it comes to vaccines or evolution or whatever you're going to deal with in life. And this is one of the things that we try to instill in the students.
It's important. It's important. I think virology should be part of the core because I think it's so important.
Mm-hmm. It's important, and it's not easy. So even, for this symposium and then the mini meeting where, as Dustin said, we locked ourselves away in the room, but we were coming at this from a lot of different angles, and it was a pretty ambitious topic. I would say the field is pretty ambitious, right? What are some of the genetic basis or vice versa of social behavior? And this is a frontier of science, literally, where it's in its infancy. You're trying to get a handle on these complex traits there's a lot of variables at play. There's interactions at play. And so if or what are the genetic contributions is not easy. And so there are a few footholds or toeholds, some of which, the folks that were in the room had discovered. But these conversations, I would say, weren't, it wasn't just sort of patting each other on the back and saying, oh, here's what we're going to write. It was kind of heavy slogging. And I think in the end, what you get is, as Dustin said, this sort of call to arms or a sense of like, what are maybe some of the big questions? What are some of the technologies? But it's still wide open. And there's massive opportunities for folks to come up and grapple with some of these ideas or new approaches and see where it might go. This is just very much, I would say, an introduction. None of the chapters have really been fully written.
Yeah, I think one of the big exciting things is that, in this world, we create genomes of a species, but, from one or a handful of individuals. And now the goal is we can sequence the genomes of many individuals within a species, and we can look at within species variation and compare that to among species variation and really get at these questions. And that's going to be sort of the next frontier, right? So currently we can do that by looking at transcriptomes, by looking at all the messenger RNA that's sequenced to look at gene expression, but we want to actually get at the genetic code more directly. And I think that was one of the big take homes for me from this paper. So this was trying to lay out, okay, if you can start to sequence these genomes, what can you do with them? And it's a first pass at that, I think. But I think once scientists start doing this, there'll be room for more collaboration with people in different fields who think about this, in typically more model organism settings.
Yeah, I agree. And I think one of the things I took home too was that, it's always this reminder about sort of, picking your system and the ones that, there's a reason that the insects are, out in the lead. So the honeybees being a great example of, a long tradition of research on eusociality. And then even though we have now honeybee genomes and we have all kinds of transcriptomics data, sort of the realization that it's a great start, but it's there's a ways to go when it comes how to interpret that data. Is this really the right data to grab? There's that old sort of politically incorrect saying of the drunk searching for his car keys, and he's looking under the under the spotlight, and that's the only place they're looking, where the keys could be anywhere outside of the bar, kind of a scenario. And so, anyway, we've got a few streetlights, and more and more powerful streetlights, but so much stuff that we're just starting to glimpse at.
And it's exciting because some of the people in the room then and subsequently have thought about what the genomics of studying, say, bees can tell us, and they're looking at social deficit disorders in bees and finding a lot of the same genes that underlie those are genes that pop up related to autism in humans, right? And so hopefully there'll become more excitement for this comparative approach across different organisms. It'll have implications for human health, but also it helps us understand the life on Earth a little bit better, and thinking about how such diversity evolves, and then when it evolves, how it feeds back on the genome and creates even further evolution.
Yeah, I agree. And then also this notion of picking new systems where the kind of behaviors or the kind of phenotypes that you're exactly interested in are the ones that you can sort of plumb into. But that's not easy either. And actually that maybe is a good pivot into your shrimp systems, Dustin. So one of the things that was really exciting, Vincent, was hanging out with Dustin a little bit during this meeting, and a bit afterwards, was to go to his lab and see some of these shrimp. Actually, I think at that point, I can't remember if your colonies were running or you were showing me some that were preserved for. I
think they were preserved at that point.
Okay, but, yeah. But even describing that and why you were chasing after these shrimp, I was convinced that this is a cool system. Maybe take us through that. Process of.
I read about these shrimp when I was a grad student, probably in my maybe third year of graduate school. And at the time, I was studying social behavior in birds, in these starlings in Africa, and they have one of the more complex social systems of really any vertebrate out there. Complex groups with multiple breeding pairs and multiple individuals that help the breeding pairs raise offspring, and they defend a territory together. That type of system is relatively rare, but structurally, it's similar to the way traditional human societies have been set up. And I read about the shrimp species that had a very similar social system. There were multiple reproductive queens. They lived together in a sponge, and this really intrigued me. And at the time, I was also thinking comparatively across starlings, where there's differences in social behavior. Some systems just have a single reproductive pair and helpers. And then in the shrimp, we see the same thing. We see colonies with a single queen and dozens or hundreds of workers, and some with multiple queens. And so this system had a lot of parallels to what I was thinking about in birds. But at the time, I thought it would be much more tractable because they're tiny. They're only the size of a couple of rice grains. We could potentially keep them in captivity. And so I started exploring this as a potential system for a postdoc and further, and then started to develop this. And the guy who discovered this was my collaborator, and still is a collaborator, Emmett Duffy, who's now at the Smithsonian, and he discovered these only in 1996. And he was doing sort of a taxonomy. He's really an ecologist, but he was doing some taxonomic work at these shrimp. And it hit him after collecting many of these colonies that there was only one reproductive individual. And it seemed like an ant, like a eusocial ant, as E.O. Wilson would say. He was inspired by some of that stuff. And so he published this paper in 1996 with that observational data, a little bit of experimental work, and some preliminary genetic work showing that they are eusocial, like ants, bees, and wasps. And so, that sort of drove us this direction. And I've become really interested in sort of this idea of comparative social evolution, thinking about similarities in vertebrates and invertebrates, and then similarities and differences within, say, birds or within, say, shrimp. And the shrimp system is nice because there's about 45 species in the group. They all live in the Caribbean. In this group, they all live inside sponges. So they're the sponge. They feed on the sponge, and the sponge protects them from predators. And then you see some species where there's a single queen and some with multiple queens, and then some species where there's no social behavior. It's just a male and a female pair living together, and some where you get multiple pairs living together in the same sponge.
And you write that it's the only genus where this cooperative living has evolved? In the marine environment? In the
marine environment, as far as we know, this is the only type of complex social behavior.
Well, there might be some out there we haven't found, right?
There probably are at deeper depths and things like that.
Yeah. And they're called sponge-dwelling snapping shrimp, right?
Yeah,
what's the snapping from?
They are synalphiid shrimp. That's the genus, and they're part of the alpheus shrimp that have these large snapping claws, and so they use these for defense. And so, the story goes they make this snap with their claw. You can hear it. So when you go snorkeling or scuba diving on the reef and you hear a lot of crackling, it's either parrotfish crunching on the coral or it's these alpheid shrimp snapping. And the power that they make out of this is intense. And so it forms this cavitation bubble that the physicists have said, create as much energy as you see like coming out of the sun. I mean, it's this massive amount of energy. We've seen the shrimp if they snap at an intruder, they'll kill it almost instantaneously with this big.
So they have one big claw, is that right?
One big claw on one side, and then on the other side they have a smaller claw with a little rake, and they use that rake to scrape the surface of the sponge, and they eat the bacteria that grows on the sponge.
Wow. And they're pretty colorful, right?
They do. They vary in color across the species, from yellows to browns and blues.
And you have colonies of these in your laboratory now.
We have at times. It's hard. We can't get the sponges to live in captivity.
I see.
That was my plan when I started a postdoc at Berkeley, and everybody laughed, and we tried for a long time. But instead, we build what look like ant farms that kids would use. We take aquarium sponge and then we sort of glue it on a piece of black plastic, and then put a piece of glass, and they'll live happily in there. The trick is it's a little bit harder than with insects because you have to keep water flowing. You don't want the conditions to go anoxic, so you have to have a little pump. But we can, if you feed them properly and you have enough water flow, we can keep them in captivity for a year or more. That's as long as we've tried.
And so when you do start to do that comparative view, it sounds like this behavior, this eusociality it wasn't just sort of a common ancestor that did this once, but this has come up several times. Is that true? What's the evidence for that?
So we've sort of built a phylogeny, sort of a molecular tree of this group using DNA and also morphological characters. And then my collaborators and I have been working on this, for 20 plus years. And so we've sampled many islands along the Caribbean and collected what we think are most of the species. And of the 45 or so species, this eusocial behavior has evolved at least 3 or 4 times independently across the group, and we try to figure out why. And then there's the eusocial species, and then there are species that we call communal, where you've got pairs living together. It turns out when you look at the genetics, these tend to be unrelated individuals. The pairs in the group, the male and the female, are unrelated, and the pairs are unrelated to each other. And so there are groups of unrelated pairs. And then you've got these eusocial species, which turn out when you look at the genetics to be family groups, basically. And then you've got other species that are just a single sponge there'll be a pair. What's kind of interesting about this group is that within a sponge, you might get multiple species. You could have a eusocial and a pair living in a communal, or communal and a eusocial. You never get 2 eusocial, interestingly, or 2 communal. We don't know why, but you get these groups of species that live together. Some of the species tend to live in only one or a few sponge hosts, and others can live in up to 14 different types of sponges.
Yeah, it's really wild. Very different than the sort of beehives that we might think of. I can't think of cases where you'd have this sort of intermingling of different lifestyles in a sense, or different sort of life histories.
Yeah, and they're completely dependent upon these sponges. If they swim out in the water, they get picked off by fish pretty quickly. And they can't. And the sponges are toxic to fish in some species, and so that may influence which ones they can use as hosts and which ones they can't.
What reason would they be swimming away from the sponge?
To disperse.
I see.
And find mates or start new colonies would probably be the main reason. We don't know much about the dispersal; it's hard to study, but we do know that when we've seen some, they tend to disperse on lunar events when it's not much of a moon there, when it's dark. Then you get like a mass spawning event in some species where they all eject out and probably go to find new sponges.
So this is a nice phylogenetic tree in the paper, which we haven't actually mentioned, Nels. Maybe we can do that.
Good point. Let's do that. So this is another review, but this one has launched. I think you can get it. Yeah. So the title is Social Transitions in Sponge-Dwelling Snapping Shrimp, and this is in
Current Opinion in Insect Science.
Current Opinion Insect Science.
Which is probably the first time I've read a paper in that journal.
Me too.
You and me both, to be honest.
It's for a special issue on major evolutionary transitions in social insects, I think. And we're the outlier because we're not insects.
You're not an insect. So, and then the authors, of course, are Dustin and then Solomon Chalk. Is that a grad student? Postdoc?
He's a postdoc in my lab.
Postdoc, yeah.
Anyway, there's a great phylogenetic tree showing all these species that you've collected, I guess, right? And they're labeled pair, living, communal, eusocial. And then you have high sponge volume, which means they're in sponges. And what does host generalist mean?
So we were interested in sort of testing sort of the ecological correlates of what might explain social evolution in this group and what might explain these social differences. And so we're looking for sort of trying to get at different aspects of their ecological niche. And so they're sponge dwellers, they're obligate sponge hosts. And so some species, as I said, will occupy maybe one or maybe two sponges only, and some, and those are we call sponge specialists, and some are sponge generalists and can occupy up to 11 or 14 or 15 different sponges. And so that was one categorization. And then some species, so some of these species live in sponges that you'll see when you go snorkeling, the big free growing sponges. Other species live in encrusting sponges, which I like to think of if you go for a hike in the forest and you find a log and there's, fungus growing on the log or between the branches. That's kind of what these encrusting sponges look like. And so these shrimp actually do best on disturbed reefs where there's a lot of dead coral rubble, and then the encrusting sponges grow in the interstitial spaces between the dead coral rubble. And so some species live in very small sponges like that, and others might live, in sponges the size of truck tires or something like that, which are very big.
So all of these species are sponge dwellers in this chart, this phylogenetic tree.
Every one of the species in the group is.
And what does it mean, high sponge volume? There are a lot of
large sponges, basically, versus small sponges.
How big can these colonies get, Dustin?
Depends on the species. So one of the species that sort of drew me to them, that has these multiple reproductive queens living together, lives primarily in these sponges that can vary in size from about a baseball up to a truck tire. Something in the size of a baseball might have, 12, 15, 20 individuals. A basketball might have 50 to 100. The truck tire size one, we've never dissected a full one, partly because these sponges could be thousands of years old and we don't want to do that. And also, but there could be hundreds of individuals in some of those colonies.
And what's the current state for that tree? What's the current state of sort of genetic information? So you used some genetic information across all the species to get this relationship. How are things going on the genome level, genome analysis?
Well, one of the things that we sort of discovered sort of serendipitously, but I guess stepping back, one of the things in my postdoc was to look at the genetic structure of these different species. And this was back in the days of microsatellite markers. So these are sort of di or trinucleotide repeats that are across the genome, and they sort of mutate relatively quickly, and they're good for parentage studies or kinship studies or population genetic studies. And so I started developing these while I was in grad school, before we even started working on this. And it was challenging because, most species you'll have either you'll have a copy of the genome from mom and a copy of the genome from dad. And so you can either have, for a given microsatellite locus, you'll have two alleles, and they could be the same or they could be different. So when you sequence these, you'll get 1 peak or 2 peaks. And we did this for the shrimp. We would get 3 peaks or 4 peaks or 6 peaks or 8 peaks. And this was puzzling for a long time. Kept thinking I was contaminating things or doing all sorts of things. And it turned out that I went up to give a talk at UC Davis, and I was talking to a colleague there, and he said, this is a signature of polyploidy, it's genome duplication. Mm-hmm. Turns out we don't think they're polyploid and they have fully duplicated genomes, but there's a lot of gene duplication in these species. And so we sort of knew that. It complicated a lot of the genetic studies we're doing. But then some of my collaborators were working on getting estimates of genome size across crustaceans. And we discovered in these Synalpheus that they have huge and widely variable sized genomes. So some species, this is looking at the DNA content in a cell. But it approximates to what we would think about in terms of gigabases of a genome size. And so some species vary from about 4, which is on the order of the size of the human genome, somewhere around there, a little bit bigger, all the way up to about 20. So some species, have a 5-fold difference in genome size across the species. And it turns out that that the more social, the eusocial species tend to have larger genomes than the communal breeding species, than the pair living species. And so this is what actually spurred a lot of this working group to try to figure out why this is. Why would you expect to see a positive relationship between genome size and social behavior? The only other data that I know of still, and at the time, was from bees. Some of the work by Karen Kapheim, who was also at the workshop, is the opposite. The eusocial species tend to have smaller genomes. And so that got us thinking, well, what's the pattern if we look sort of more broadly and think about this? And there's hypotheses to explain it one way or the other. And some of those are in that paper. And so we've been working on this. Solomon's been working on this for his postdoc for the last few years. And he's almost there. He finishes in a few months, and he's almost got the answer, but it's probably due to the proliferation of certain classes with transposable elements and SSRs like microsatellites.
But, the TEs, the transposable elements, have coding regions or not?
That's one of the questions to be determined. Yeah. So we're going to try to do some transcriptomics to get at that. It's hard to get at transposable elements in crustaceans because we don't know a lot about transposable elements in crustaceans. So we don't have the databases to blast against. There's a couple of crustacean genomes that have come out, in the last year or two. So we have some evidence, but there seems to be a lot of, shrimp and maybe Synalpheus specific group classes of TEs that we're trying to sort out.
Are some of them retrotransposons? Yes?
Yep.
I'm thinking, Nels, there must be some interesting viruses in these shrimps.
I bet.
Do you know anything about the microbiome of the shrimp?
We haven't fully looked. Solomon did some stuff in his PhD to look at the bacteria, because they're eating that from the sponges. So he was trying to match up sponge phylogenies versus shrimp phylogenies and see what they're eating. But nobody's really done that. I always say, I often give a talk, and at the end, when I speak about shrimp, I have a slide of an ant with like a do not enter sign, because ants are fascinating, as you've said. But there's hundreds, if not thousands, of people that study ants. There's probably like 4 people in the world that study these shrimp. And we all sort of do it as side projects now. And it's a rich system. And so we're very open to anyone who wants to study viruses or microbiomes or behavior or genomics in shrimp. Come get in contact.
Well, let's talk about viromes. I'd be interested in looking at that. If you have nucleic acids from, we can easily go through it.
We got 50,000 shrimp downtown.
You have DNA. You have DNA and RNA as well.
We do, yeah.
We should talk, because I'm really interested in different viromes. Although I'm at a medical school, I probably shouldn't work on shrimp, right?
I disagree. I think you should. I think this gets to some of the idea of bringing these collisions together.
But this is how you find new viruses and things too, right? So somebody contacted me with some of the transcriptomic data we published on the starlings. Said he found what looked like a new avian virus, and could we send him some more tissue? So this is someone in Europe, and we sent him some samples, and there's a whole bunch of new avian viruses discovered in these African birds. So the shrimp, who knows what's in there?
Well, we're talking about behavior manipulation by viruses. It would be amazing, right? If viruses drive eusociality, right?
It would be very cool.
Or vice versa, right?
Yeah, no, I just think this also is a great illustration. And I think, Dustin, you're describing it really fairly as well, that this is not like simple, like the years that have gone into this, the like literally getting your hands dirty, the, sort of romantic notion of you're scuba diving all day or snorkeling, but you're actually mostly kind of making your way through coral rubble on a dock bent over. You start to look at the genomes. That's, you get curveballs there. They're massive in some cases. How do you, and I think it's easy, in science fields and sometimes to celebrate the successes. So we've had Nicole King on to talk about choanoflagellates on TWiEVO, and she can tell a story that has some similar echoes about all of the challenges of building that system from scratch over decades. So it's really worth it on the one hand, because the biology is amazing as a system. It's amazing, but to really give it that honesty of what it takes to put it together and how it's going to take team efforts, and not just to have 4 labs around the world, but for sort of ambitious and interested, skillful people to kind of come in and bring their own curiosity into the conversation.
Definitely. When I was a postdoc naive in the sense that, we can get this system to work, right? It was behaviorally fascinating, but the biology was pretty unknown. So, I mean, I spent a lot of time doing very mundane things like realizing when females become receptive. So they tend to molt. The shrimp tend to molt on the lunar cycle, and that's when females become receptive and males guard them. And so that's sort of the important time. If you actually watch them, they don't really behave much. We discovered that a lot of these species are intersex or hermaphroditic, and they seem to probably switch from one sex to the other. We don't quite know why, but we think they switch from male to female, and there's a lot more competition in females. And so, but to figure this out, we didn't discover this, and another set of shrimp biologists discovered this. But to sex a shrimp, you have to use scanning electron microscopy. Wow. And so one sex has a certain opening under one set of walking legs, and the other sex has a different size opening under a different set of legs. And so and some species, they've got both. And so we think they're both. And so it's challenging to do experiments. It's challenging to even, if you want to study social evolution, you kind of got to know how many males there are in a group, how many females are in a group. And that's really hard. You can also look at the sperm and the ovaries, but to do that, you have to preserve them a certain way. So we have, 50,000 specimens that Emmett Duffy and his collaborators have collected over the years sitting in the lab down on the Morningside campus. But you can't really sex those. You need to preserve them a different way if you want to look at the sperm and eggs. And so, you take for granted, I guess, the basic biology, not just in model organisms, but in species that are as well studied as shrimp. As ants or bees or wasps and something like that when you compare it to the shrimp. And so there's a lot of biological diversity out there that warrants more study. But you gotta be patient. And so it was probably a risky project to start as a postdoc and as an untenured professor, but I also work on birds. And so that was a more productive system as I go.
Mm-hmm. Is this hard to get financial support to do this shrimp work?
We've gotten grants from the National Science Foundation. I think people find it as a biologically fascinating system, and when you want to compare it to better known social systems, like the insects and things, people are willing to fund it. But you're not going to get funded at NIH or something like that, unfortunately.
This is not medical stuff, but that's okay. I noticed you have support from Simon's Foundation, right? That's an interesting foundation. I worked with them for a while, and I know they're mostly interested in math and physics, but they have been branching into life sciences.
Yeah, they've been funding some postdocs. The one who's doing the shrimp work actually with the genomes is funded by them through the Life Sciences Research Foundation, and they've funded two other postdocs in my lab studying birds. But they've I think the funding is almost done funding sort of a comparative socio-genomic grant at the University of Illinois. So they're interested in these ideas to some degree.
Do you have a contact there at the Simons that you work through with these?
I wish. No, I don't, unfortunately.
Because my colleague next to me is Steve Goff. His wife, Marion Carlson, works for the Simons.
Okay.
And she's always looking for new projects to fund. I know she was really interested in microbiome and some marine projects as well. So you might contact her because this would be something that they're looking for unusual stuff that NSF and NIH are not supporting, where they can make an impact.
Yeah. Crustaceans are interesting. I mean, I think these large genome sizes that we see in crustaceans are actually pretty common. Mm-hmm. So, the little brine shrimp that you probably had as a kid, Artemia.
Sea monkeys.
They have something like 200 plus chromosomes, right? They're huge. And so, they're going to have large genomes. And so when you think about when you guys are interested in sort of genome evolution and virus evolution, we don't even know much of the biological diversity under the seas, but they've got probably huge and scrambled genomes with lots of transposable elements. And there's probably all sorts of viruses in there that nobody's even looked at.
That's right. Even among fish, just even in the last year or two, some bigger metagenomic surveys of fish viruses are among even just RNA viruses, revealing basically every class of RNA viruses we know on land also exists in fish. The fact that we are, 2018, 2019, and just learning this now, I think shows again how little we know about aquatic environments.
You just have to look, right? I mean, it's most environments have never been explored because there's too much for for the number of scientists that, there are a lot of us, but not enough to hit everything. You can always, even people. I was just reading a paper before you got here, brand new virus in the respiratory tract of people. It's been there forever. We just never found it.
Amazing.
And so you can go to all these environments and find. That's why looking for viruses is a lot of fun, because you're going to find something. Always.
I'd say also a common theme here that is this idea that for stuff that scientists work on, like the shrimp system, that oftentimes you'll be funded for other stuff, the birds or, other projects, but you kind of bring it along on the side, kind of almost secretly funded or fund it through other channels almost until it gets its moment in the sun. And that's very common to a lot of PIs that running their labs that do just that in all fields. Sort of a common theme, actually. It takes a while. Having the flexibility and the space to do this and to support this more explicitly, I think, is a potentially major growth area of science.
Yeah, I think running a lab is almost being an entrepreneur, right? You not only have to go out and secure the funding and get the right people, but you got to come up with the ideas, and you got to have some risky things going on and some more established things going on. And, I think the exciting labs are the ones that are able to take those risks. And, they're not all going to pan out. But if they do, it leads to important discoveries a lot of the time.
So how many people in your lab are working on the shrimp project?
Right now, a postdoc for another month, and then,
Solomon,
and then he's going to start a job. He's going to take another postdoc for a year, but he's probably going to start a job he's going to defer and with a heavy teaching load and doesn't have a graduate program. Emmett Duffy runs a major program at the Smithsonian now on sort of seagrass ecosystems, and doesn't have a lot of time for shrimp. And then one of our collaborators, she works on crustaceans more generally, just found out she got a big NSF grant to work on something else for the next few years. And she's at a small school, doesn't have a major graduate program either. So I don't know who's going to be working on shrimp, and probably I'll have to get back to it. I'll have to write a grant, I guess, to follow up on some of this genome evolution stuff. But no, we'll keep it going. It's too behaviorally interesting and genomically interesting. I think we can't sequence the genomes fully yet. We do, sort of half X coverage, and we do things and we don't assemble them. But maybe someday we can, sequence this 20 gigabase genome.
If we can get some virus preliminary sequence, maybe we could write something.
Definitely, yes.
Push that forward.
Yes, I'll give you some samples this week, and it would be fun to talk.
All right.
Mm-hmm. Cool.
So now, should we start to wrap up so that we can let Dustin go at an hour and a half like we promised?
That's right. Exactly. Got to hit that New York City traffic.
Yeah.
Shuttle back right in the middle of into the teeth of commencement.
Hopefully it's done. I think everyone's moving out of the dorms now. They don't give them much time.
Yeah. That's right. Yeah. Let's move into our science picks of the week. So what we do, Dustin, is try to come up with something that kind of caught our eye in the last month or so. It doesn't have to be directly related to science, just something interesting. I had one actually. I'm doing a last minute switcheroo here, Vincent. So Vincent and I will do our picks. Yeah. And if anything comes to mind, Dustin, feel free to throw in on one, but no pressure either way. So I was going to talk a little bit about this new beekeeping technology called the flow hive, just kind of given the social insects and the sort of topic here. But I made a last minute switcheroo. So talking about in sort of the opening there. About Cornell and Thomas Eisner, his book For Love of Insects. I'm choosing that as my science pick of the week. So I've got the Amazon link here, and I think I have a copy of this. I have to confess, I'm still just starting to dig into it, but I think a really beautiful story that gets to sort of this giant in the field that. You were describing, Dustin. And so a book that certainly hits the science notes, but also I think captures a broader audience. I think it's worth a look.
Yeah, I have to look at this. I haven't, but I hear it's great. Mm-hmm. So Nels, based on our discussion, I don't know, last time, we talked a little bit about Svante Pääbo, remember?
Yeah, that's right. The guy who does a lot of genomic sequencing in archaic human populations, so the Neanderthals and others.
So I bought his autobiography, and cool. I read it, and it's just great. It was my pick on TWiEVO a couple of weeks ago, but I highly recommend it as well. I don't know if you've read it, Nels, but
I actually haven't. But
it's really great.
Also on
it's
my
really
list.
great.
Yeah.
He describes his work leading to the sequencing of the Neanderthal genome, right? And he's a great writer. It's really suspenseful. He tells a great story about all the technical hurdles. These old bones, most of the DNA is crap. Contaminants, people touching it. He tells this story of going to a museum and trying to get some Neanderthal bones. And he asks the guy, "Has this been coated with varnish?" And he picks it up and licks it. He goes, "No." And then he's freaked out because, oh my God.
Coated with modern DNA.
So they have to figure out ways to find like the 1 to 3% of the DNA that's Neanderthal, right? Anyway, it's good. I highly recommend it. But it's not my pick today. I already picked it. I don't want to do it twice. My pick is an article in Ars Technica. It's about this outbreak of HIV/AIDS in Pakistan.
Oh, yeah. I think I heard about that.
Like 600 people have turned positive, and it looks like rogue doctors and unqualified people practicing medicine are behind this. They're using contaminated syringes, and they use this because they're cheaper, right? Reusing syringes. And this is really sad, but the amazing thing here is this is probably how the HIV emerged in the first place in Africa in the 1920s by European countries moving in, colonizing Africa, starting healthcare centers, and reusing needles. And that's probably how they spread the infection. So here we are. We think we're so advanced, but we're still doing similar things. Reusing needles is just really bad. So that's an unfortunate thing there. But it's an Ars Technica article about it.
Yeah, good to keep an eye on these
by
things.
Beth Mole.
And
anything you'd like to share with us, Dustin? A good book you've read lately?
I've got my summer reading list. I've got 2 books that on sort of social behavior that are on my list that I want to read. Both just came out in the last month. One by Mark Moffat, who's based in the city here, and it's sort of about ant societies and relating them to human societies. And then I think E.O. Wilson has a new book. E.O. Wilson was actually his PhD advisor on a similar topic. They both came out at the same time. So those are, I have one already. I'm going to pick up the other one, and on my travels this summer, I hope to read those and see what people are thinking about, again, on this comparative social evolution view, but taking this ant perspective and relating it to, in this case, humans.
Cool.
Very, very cool. So that first one by Mark Moffat, I think, is this called Adventures Among Ants?
No.
Or that's older.
He has a new book that just came out in April. I think it's The Human Swarm, maybe.
Okay.
And I don't remember the title of E.O. Wilson's. I've just seen the review on it.
Yeah, Human Swarm.
Yeah.
That's it. Here it is.
Cool. We'll put some links to both.
How our societies rise, thrive, and fall. And they do fall.
Yeah. So haven't read them yet, but it's they piqued my interest, and now that classes are done, maybe I'll have some time to read. Maybe.
Yeah, 2 good picks.
All right. That is TWiEVO 43. You can find it at microbe.tv/TWiEVO, and you probably listen. If you go to microbe.tv/TWiEVO, you get all the show notes, the links, and everything that we talk about, but you probably listen on some kind of podcast player on your phone or your tablet. And whichever one you use, we'd like you to subscribe. That way you get every episode automatically, and it's free, of course. And that way we know who's listening, how many people are listening. So please subscribe in your favorite podcast player. And if you like what we do, consider supporting us financially. You can go to microbe.tv/contribute. And, for example, you could give us $1 a month over at Patreon. And that supports all the shows that we do at Microbe TV, TWiV, TWiEVO, TWIP, TWIM, and Immune. It's like 10 shows a month, 4, 5, 6, 7, 8, 9 shows a month for free. All good science. How can you beat that? Less than a cup of coffee. $1. I told Dixon yesterday on TWIP, I don't think you can even buy a cup of coffee for less than $1 anymore.
Not in New York.
But you can get 9 podcasts.
Not in Salt Lake.
Our guest today has, and of course, TWiEVO at microbe.tv for your questions and comments. Our guest today has been Dustin Rubinstein from Columbia University. Thanks so much for joining us.
Thanks for having me. It was great.
I'm glad you came.
Yeah, thanks, Dustin.
Appreciate the time. Nels Elde's at Oh, by the way, Dustin's on Twitter. Dustin Rubinstein, all one word.
Dust Rubinstein, I think.
Is it Dust?
I think so.
Okay, I'll take out the IN. Dustin Rubinstein. I looked actually at the paper. It's funny, the paper that we talked about, the Symposium paper, has everyone's Twitter handles.
Yeah, the journal asked for that, actually. We went there originally, and they said it's not on the proofs, so I don't know what they're using it for. Maybe just to tweet out at the end. They took it off the proofs.
Really?
Oh, interesting.
But they specifically asked for that.
Right, yeah.
Not everyone has a Twitter handle of all those co-authors. Maybe you can guess which ones don't. But
yeah, that's right. And by the way, congrats on that paper, Dustin. Both you and Hans. That was to host a really inspiring symposium and then follow through from that micro meeting to actually publishing something. That's pretty unusual.
Thanks.
So
well, you guys wrote a lot while we were there, and we took a while to get to it. But I'm glad. Hopefully it'll get people thinking and spur some of this new research in these new directions.
Yeah.
Nels Elde's at cellvolution.org. He's also on Twitter. Early Bird. Thanks, Nels.
Thank you, Vincent. Good to be in touch. Semi in vivo, at least through video link today.
Yeah, today we were looking at each other, which we usually don't do, but it's actually kind of nice, right?
Yeah.
Maybe we should do it more often, especially since you're recording locally and it doesn't affect our bandwidth. Hey, it's very good video. Skype has gotten better.
True.
And you're in sync. A lot of times your lips don't match the sound, but you're synced. Let's see.
Pretty good. And luckily I was wearing pants today, so it was all.
Early Bird. I'm Vincent Racaniello. You can find me at virology.ws, PROFVRR on Twitter. Music on TWiEVO is by Trampled by Turtles. They're at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.
How did you guys get hooked up on this together?
Nels is a virologist, and I am too, and we, go to the same meetings. In fact, was it 2015? We were at ASV in London, Ontario. Is that right, Nels? So he got some award at this virology meeting, and I presented it to him. And we were at the bar that night, and I said, you should do a podcast. You'd be good at it. And he said, let me think about it. And then, because he's very cautious about using his time, Yeah,
it's a big commitment.
Which I understand. I'm out of control, but, and 6 months later, he said, yeah, I'll do it. He said, we'll do it once a month. So we're on episode 43 now. Wow. Even though we call it This Weekend, it doesn't.
Yeah. And how many of these do you do? You do it more often than once a month.
So I have 5 different podcasts. The first one I started was This Week in Virology, which is weekly, and that's at episode 550.
Wow.
And then we do This Week in Parasitism. I have a colleague here, 2 colleagues here I do that with. That's about once or twice a month. I do microbiology once or twice a month, and we just started an immunology podcast, which is once a month. And all of them have different hosts, so they're really nice to give me time, right? But people love it.
Great.
We get great letters, and we have a really engaged, many thousands of people listen, right? It's not just 10 or 20, but thousands of all sorts. It's amazing. We get PIs. We do a paper, and the PI emails us, I heard my paper on your, Right now, am I telling them the truth?
It shows up on some of the Twitter conversations that kind of echo along after our episodes. It's really fun to see people respond and correct us when we inevitably get some key details a little bit scrambled. But that's really great.
Yep.
And we've had a few guests, similar scenario. Josh Drew came by.
Okay.
Do you guys connect at all? Dustin, you and Josh?
He used to be in my department, but he is no longer in my department.
Gotcha.
We've had Hoekstra, right? Who's the co-author on this massive paper you guys are all on.
Yeah. And
then we have Corey Moreau, right?
Yep.
We had her.
Sweet Chance. Yep.
So Nels is the go-between. He knows everybody in the field because it's not my field. So that's the thing on every podcast except virology. I get people who know the field.
Yeah, so you get to learn a lot.
And I learn a shitload. I'd never read any of this stuff, right? But if I had done it from day one, I would have been a better scientist.
Yeah.
For sure.
Yeah.
But it's hard to keep it up.
To be honest, I also wouldn't read a lot of this stuff. I mean, even within our fields, right?
I know, and
different cultures, right?
Yeah.
I find it hard to find time to read papers, widely anymore. My time is taken up with other things, and I download papers and I look at the abstract, and they sit there for 5 years, and I never read them. So this is a good way to force you to do it, I guess.
I think a lot of students also listen so they can hear papers discussed, and they don't have to read them, or if they're looking for something for a journal club, they get ideas in all the fields we do. So we get a lot of students listening, but we also get non-scientists.
Okay.
People who are just interested in science and they want to hear scientists talking, because there are not many opportunities to do that. Right? A lay person out there can't hear scientists talk. They can't go to meetings, so it serves a purpose. So I'm going to do this beyond my lab,
Okay. Yeah. I didn't quite realize it was so much on the papers. I should probably read that tree paper. It's been a while, but
no, it's all right. I'm sure you'll be fine. I'm sure you'll be fine. I teach a course down at Morningside, by the way. I'm there every spring. I teach a virology course.
Okay, in the biology department.
Yeah, and I just finished it.
Today was graduation. I just turned my grades in a little bit late, but
yeah, I have to get. I put my graduation senior grades in Friday, but I have to get the others in this week.
Yeah, I didn't have any seniors, luckily, but I think they're due tomorrow.
Yeah, I'll get them in. It's no problem. All right, Nels, you want to do a podcast?
Count me in.